Okay, our next talk for this morning is by Professor Mark Kurth. Professor Kurth completed his BA degree in 1976 and proceeded to graduate studies in the laboratory of Professor Thomas Hoye at the University of Minnesota, where he did total synthesis of natural products. He finished his PhD degree in 1980, and then he joined Professor Wolfgang Oppolzer's group at the Université de Geneva for postdoctoral work. Then he joined the faculty of the University of California, Davis, where he started his 10-year career. He received a long list of awards, including the Alfred Sloan Fellowship in 1987, the Eleanor Roosevelt International Cancer Fellowship in 1988, NIH Research Career Development Award in 1989, the University of California ASUCD Excellence in Education Award in 2004. He also received the University of California Chancellor's Award in 2002. Professor Kurth is currently a distinguished professor at UC Davis. And of course, Professor Kurth and Dr. Haddadin, they discovered what is called the Davis-Beirut reaction. I'm very pleased to welcome Professor Kurth. The floor is all yours.
Thank you. What is this? Do up there. Oh, how was easy.
It's working.
Great, thanks. Bilal, thank you. Thanks to everyone for getting me here for this wonderful opportunity to. Okay. Sorry. To be here and to say hello to friends that I know here at AUB and to give this lecture. It's exciting to be here for the 150th anniversary of the American University of Beirut, and it's equally exciting to be here for MJH 81, Makhlouf Haddadin 81. And I should add that the anticipation is, and counting is applied to both. So the years are piling up. The American University of Beirut ranks 5th best overall in the Middle East. And Makhlouf Haddadin ranks best collaborator and colleague. Now I'm talking to a bunch of scientists, so I know you're all thinking, well, you can't just say that, you have to back that up with something. So let me try to back these two statements up. It's the US News and World Report that did this ranking, and it was of 91 universities from 16 countries, and AUB ranked 5th, which is an incredible accomplishment. For Makhlouf Haddadin, well, that's the world according to MJ Kurth, but I would add many others. And let me show you a little bit of the data that I used to get that ranking for Professor Haddadin. I've published papers with 421 co-authors, and this is the start of the printout from SciFinder. And you'll note that, You'll note that, I have to back up, sorry. Makhlouf has, and I have 35 joint publications, and you'll also note that that's less than I have with Marilyn Olmsted. She and I have 37 joint publications, so you might think, well, it's not really fair to say best for Makhlouf then. Maybe I should say best for Marilyn. But let me also, as pointed out in the introduction, point out that Makhlouf and I have the honor of having an organic reaction named based on our work, and it's the Davis-Beirut reaction. And so in my mind, that makes it best. So thanks, Makhlouf. Now here this is a relatively recent picture of the two of us standing at the board in my office talking about the mechanistic details of the Davis-Beirut reaction. And I want to point out two things. One is when you're at the board with Makhlouf talking about mechanistic details, find the pointer. He's the man who always controls the chalk. It's just part of the game with Makhlouf. And the second point is that you might wonder, what is the point I'm making in this moment? And it's that this number, 35, needs to go to 38 so that I can say best even for bean counters. So I hope that makes sense to everyone. So with that, let me sort of turn to the title of my lecture, which is MedChem Studies of CFTR Function. And by giving this lecture today, it's an honor to tip my hat to AUB and to Professor Haddadin. So what is CFTR? CFTR stands for Cystic Fibrosis Transmembrane Conductance Regulator. It's a gene, and that gene produces a protein known as the CFTR protein. And the CFTR protein, as stated there, is an ATP-gated transporter class ion channel that transport chloride ions across epithelial cells. So that's the whole point of CFTR. Now, as a chemist, that sounds like a reasonable statement, but I have to also point out that it's a complicated process that makes that all happen. First of all, of course, transcription has to take place, where messenger RNA is produced, and then that's translated into a protein structure. That protein structure is post-translationally modified, and when appropriately modified, shipped to the cell surface. At the cell surface, the protein has to insert itself into the cell membrane. All that's fine and well, and for it to actually function as desired, it has to be properly gated. So that's a lot of things that have to happen for CFTR to do its business. It's interesting, we sit here, all of us in the audience, and myself included, doing that without much forethought on our part, and we take CFTR for granted. Let me sort of talk to you about what happens when CFTR has a problem and the kinds of issues that creates for people. I'm going to try to get through all this today. In cystic fibrosis, the most common form of cystic fibrosis, there are sort of a couple of problems. One is the protein is never trafficked to the cell surface. So no protein at the cell surface, no chloride channel. In cystic fibrosis, patients get a thick mucus that forms over their epithelial cells, for example, in their lungs, and that thick mucus is an excellent place for bacteria to grow and bacterial infections to happen. And so there's issues with bacterial infections, and we have to address that at some level. In polycystic kidney disease, which is another genetic disease, and in secretory diarrhea, the fundamental issue is a chloride channel, a CFTR channel that's improperly gated and stuck in the wide open conformation, and chloride is exiting the cell in an uncontrolled fashion. And that creates some problems. And then in constipation and dry eye, it's sort of the other problem is the channel's not properly gated, or at least in the moment, for the issue, and there's not enough chloride being secreted, and so we want to find things that in CFTR are potentiators and correctors to address these problems. In this bacterial infection, we want to inhibit the enzyme myeloperoxidase, and I'll tell you more about that. For polycystic kidney disease and deep secretory diarrhea, we want to find inhibitors that can gate the channel and close it. And in constipation and dry eye, we want to find activators that can cause that channel to open. So those are the kinds of things we're trying to address with medicinal chemistry, and I'll tell you that story then today. And the point here with this little cartoon is that CFTR is a thing, but depending upon your perspective on that protein, the challenges are different. You know, in one case I have to open the channel, in another case I have to close the channel, in one case I have to get the protein to the cell. There's a bunch of things that we have to consider, so you can imagine interesting dialogue with you and your colleagues in that regard. So for those of you who aren't American football fans, this is the UC Davis football stadium. And the point I'm trying to make here is that if you're playing football, the coach will tell you if you're down here and you got to bring the ball to this end of the field to score a touchdown and make points, the coach would say, there's a lot of turf to cover, so let's get going. And the point I would make is that if I'm going to try to cover all of this today, I have a lot of turf to cover, and I have to get going, and so I'm going to move at a kind of a happy clip. The other thing I'd like to say sort of at the start is that for the young people in the audience who are studying chemistry or related fields, you have a lot of career options that you're looking at and a lot of opportunities to go in different directions. And the invitation I would like to afford to you today is to consider synthetic organic chemistry as a career path. Because it's a career path that just, in my experience, has opened doors. Every time I turn around, there's another unique, interesting opportunity to apply synthetic organic chemistry tools to. And so I would invite you to at least consider thinking about being a synthetic organic chemist. Okay, so let's say a little bit more about this protein. It's a 1480 amino acid protein. It has 2 nucleotide binding domains, 1 and 2. It has a regulatory domain. It has 2 membrane spanning domains. There's the first and here's the second. And the properly functioning protein creates a chloride channel and it lets chloride escape the cell to the surface of the epithelial cell. If you're going to think about trying to address the problems associated with CFTR, what I can tell you, it's very difficult for one lab to contemplate all the issues and to do all of that. And so it's really collaborative science. And I want to just make that point very clear, that it's collaborative science that allows this kind of project to have a path forward. In my view, it takes a molecular biologist. That's the symbol I'm using for molecular biology. It takes a team of people doing assays and screening. That's my symbol for assays and screening. And it takes a synthetic chemist, and this is a little bit egocentric perhaps, but I like to think of the synthetic organic chemist as standing at the center of the universe, spinning out molecules that change the world. So a little egotistic, but makes the point that it's going to require input and insights from a variety of scientists to address these kinds of issues. So again, when I know at the beginning stages of this kind of work, when you meet with your collaborator for the first time, it's really almost hard to come up with a dialogue where you can both share information and talk because the perspectives are so different. So that's the challenge we face. We're all sort of staring at the molecular biologist knows and sees things, the assay team, the synthetic chemist, but we're really not being able to chart a path. But by collaborating and getting together and having team meetings and conversations, you can begin to work as a team and see opportunities that no one lab or individual could do. So I think that's kind of the point of this kind of work, is it's really a team effort that makes it go forward. Okay, so let's think about cystic fibrosis. And now this is kind of coming at it from the molecular biology perspective. Turns out there are 1,800 known mutations of the CFTR gene, which leads to mutations in the protein. And those mutations can fall into one of 5 categories. They're listed here. The no synthesis category, the protein's just not made. The block in processing mutations that prevent the protein from successfully making it to the cell surface. Block in regulation, where the protein's in place, but it doesn't function properly, doesn't respond to cell signals in the way it's supposed to. Altered conductance and reduced synthesis. So again, 1,800 mutations that are known, fitting into these 5 categories. The most common form of CF is delta F508 CFTR. And what that means is at the 508th position of this 1,480 amino acid protein, there's a missing phenylalanine. Because that one amino acid is missing from this protein, all the problems of cystic fibrosis happen for that patient. One missing amino acid. It's kind of scary to contemplate. The thought is that this mutation occurred about 52,000 years ago, and that it provided a slight advantage to the person with the mutation against cholera. They had a slightly less effective CFTR channel. They were exposed to the cholera toxin. They didn't die from secretory diarrhea because they had this slightly less effective CFTR. So that advantage allowed it to propagate into society. And it leads to the point where in today's world, in the United States, 1 in 30 people are a carrier of a CFTR mutation. 1 in 30 people. So let's say there are 90 people in the room. If this were in the United States, 3 people in this room would be carriers of CFTR mutations. Now, the other point to make is it's autosomal recessive, which means if you have a normal copy and a mutated copy, the normal copy dominates and you live life pretty much okay without many issues. So it's autosomal recessive. And there you go. If you think about the genetics of that, then if there's a father who's a carrier and a mother who are a carrier, and for the most part they wouldn't know that, they marry and they have a child, there's a 1 in 4 chance the child will have normal CFTR from both parents. Both genes will be normal. There's a 2 in 4 chance that the child will be a carrier of the mutation. And then there's a 1 in 4 chance that the child will have cystic fibrosis because it has 2 copies of the defective gene. So if you do the math on that, if 1 in 30 in the US are carriers, 30 times 30 times 4 leads to 1 in 3,700 children born in the US have cystic fibrosis, just from that math. And again, as I said, this mutation, the Delta F5 mutation, accounts for 70% of cystic fibrosis cases. So if you're thinking about tackling cystic fibrosis, of course the one to think about is this one, which is a block in processing where the protein doesn't get to the cell surface. Okay, so that's sort of the background. Oh, and I should also point out that it has all kinds of effects in the human body, but the primary effects are in the lungs, the pancreas, and the small intestine. Those epithelial cell surfaces are where the damage is really done in cystic fibrosis. Now, this is an organic chemist talking about molecular biology, so this is a cartoon that works for me. The cell has an inspection system, and it looks at that newly formed protein and decides it's good or it's not good. And in the case of Delta F508 CFTR, the cell inspection looks at it and has the choice of saying, oops, we made a mistake, let's take that protein apart and redo the synthesis, or let's ship that protein to the cell surface and get on with business. And it turns out in Delta F508 CFTR, the answer is that it decides to take the protein back to its building components and redo the synthesis so it can correct the mistake. But of course, it's following genetic code, so it keeps making the same mistake. So it's misformed, it's taken apart, it's resynthesized, it's taken apart, and you can see why it never makes it to the cell surface, because it gets caught in that loop and it never gets shipped to the cell surface. So that's sort of the fundamental problem in Delta F508 CFTR. My colleague, Alan Verkman at UCSF, is sort of the molecular biologist and the assay person, so he's the two-component person in this team, and I'm the synthetic chemist for this team. Did a kind of an interesting thing, and I guess it's because he's very bright. He take rat thyroid cells that were transfected with Delta F508 CFTR mutation and cool them from physiological temperature, 37 degrees, down to 27 degrees. And magically, the decision by the inspections team is now different. It says, okay, at 27, let's not take it apart. Let's send it to the cell surface. So that was a very interesting and enabling opportunity for us to start a project. Without that observation, I don't know how we would have gone forward. So I'll try to explain that in a second. Now, of course, you can't randomly take a person and cool them to 27 degrees C every once in a while and to get the protein to the cell surface. So what that means is if you're going to try to tackle Delta F5 CFTR, and I meant to say this on the previous slide, when that protein is escorted to the cell surface at lower temperature, when it arrives at the cell surface, it's incapable of forming a channel. So not only does it not get there because of the mutation, it doesn't function because of the mutation. So that means we have to do two things. We have to discover what are called potentiators, something that allows the channel to be properly gated to become a chloride channel. And we have to find, and that's sort of represented by this mechanistic detail. And then we need to be able to get it to the cell surface, cause that shipping to place, and that's called a corrector. So those are the two things we have to find and do if we're going to address Delta F508 CFTR. Before I really do the next step in this, I'd like to sort of put a human face on the disease Delta F508 CFTR. The first thing I'd like to point out is that in today's world, the life expectancy, if you have cystic fibrosis, is 37.4 years. And that's a life expectancy that's on the shoulders of antibiotics that work well, on surgical procedures that work well. A lot of things have to happen to reach 37 years of age with cystic fibrosis. So it's a very devastating disease. In the not so distant past, people died as children with cystic fibrosis. So those advances in medicine have taken it to 37.4 years. Introduce you to Tara. Tara's a young lady who lives in Davis. She does fundraising and donates money to my group. She has cystic fibrosis. She has Delta F508 CFTR. She's in the record books as a mom with cystic fibrosis, Delta F508 CFTR, who successfully carried twins to term. That's a very rare accomplishment for a mother with cystic fibrosis. She did it. This was a picture taken with her twins and her husband about 10 years ago on the twins' first birthday. So I should point out that the father has normal CFTR. She has cystic fibrosis, which means these children are now carriers of cystic fibrosis, but they don't have any really symptoms of the disease. So this is now a picture of Tara taken this past spring, about 10 years after this photo. And it shows her on her first day where she was allowed to leave the hospital in a wheelchair after a lung transplant. So you can see it's a very devastating disease. Here's a young woman who is, she's incredible how she manages the disease. And yet she needed a lung transplant. She celebrated her 38th birthday, had a big life expectancy party on her 38th birthday about a year ago. So it's quite impressive. All right, so now let's think about the assay team. If you're going to start a project, you have to develop an assay that allows you to look for solutions. And the assay that Alan's lab came up with is shown here. He took these rat thyroid cells, transfected them with CFTR mutation and green fluorescent protein generation tools. So these cells have mutant CFTR and they have green fluorescent protein. He then cooled those cells to 27. That causes the cells to be shipped to the proteins to be shipped to the cell surface. And then you can add a test compound to that Petri dish and ask, does the test compound open the channel? Again, now what we're looking for is a potentiator, something that causes the channel to open. And if the channel opens, the cells are bathed in iodide, aqueous iodide. The iodide can enter through the open channel and quench the fluorescence. So there's an assay. You can look for a drop in fluorescence if the compound is serving as a potentiator opening the channel. So that's how the project starts from his end. Now, you might be sitting there thinking the same thing I was thinking during the first conversation with Alan about this is, wait a minute, Alan, we have the cart ahead of the horse. What good is a potentiator if we haven't gotten the protein to the cell surface? And Alan said, trust me, Mark, this will work. It's okay. We have to develop a potentiator first. So I said, okay. And away we went. And what Alan did was to get his hands on large collections of compounds, 50,000, 75,000 compound libraries, and do this assay one compound at a time, looking for compounds that were potentiators. And he finds what really is a needle in a haystack, a compound that does open that channel. The way it goes then forward from there is I get a telephone call from Alan, followed by an email with the structure attached, and he says, according to the barcode on that vial, that compound, that's the active, the potentiator, should have that structure. So the first thing my lab does is looks at that compound, and Alan says, so I need you to verify that that's indeed the structure. So we have to come up with a synthesis of that compound. We have to make it. Now there isn't a protocol for that. It's been made by someone, usually in some large company setting. It's part of their library of compounds. They share those libraries with us because cystic fibrosis is an orphan disease that the company really can't address because there's no profit margin in. So they'll share their large collections. So my lab has to start by thinking of a way to make that compound. So we start doing synthetic chemistry and set out to make that compound. Once we make the compound, we send it to Alan. He verifies that that's the actual hit compound. And then the question becomes, can we make that a better potentiator? And that's a matter of doing modifications of the structure, changing things and see if you can make it better. So it's kind of a random process in a way, but that's what we get off doing. That's sort of what I would call left brain process. In other words, you have a script. This is the lead compound. Can I make it better? How can I think about making it better? Well, I can change that from an allo group on the amine to some other group, and I can just try that and see what happens. So we start doing that using, I would call sort of our left brain. Now, at the same time, the right brain is engaged, and it starts thinking of, this is a quinolynone. There you go, that core structure. How could I make quinolinones? And so you start doodling in the lab and at the chalkboard, and you come up with ideas. Oftentimes those ideas have nothing to do with making analogs of this compound, but your right brain is a pretty powerful part of your head, and you find yourself intrigued by just synthetic challenges. So in this case, we got to wondering if we had an isoxazole ring with an orthonitrophenyl at the 3 position and an orthonitrophenyl at the 5 position, and we did a reduction reaction. We could imagine reducing the nitros to amines. We could imagine reducing the isoxazole to a ketoimine. And then we could wonder, once you got to here, would the blue nitrogen cyclize onto the green imine to make a quinolinone, or would the red nitrogen cyclize onto the ketone to give an aminoquinoline? So it's kind of an interesting question. And so we decided to walk down that path. How do you make that? It's a simple 1,3-dipolar cycloaddition of a nitrile oxide to an alkyne, delivers the isoxazole heterocycle. Treat that with iron, and it reduces all those functional groups to give this guy. And interestingly, it only gives the quinolinone product. None of the quinolinamine is formed. So that's kind of an interesting observation. We later discovered that if you add a ketone to the reaction vessel prior to workup and a little bit of acetic acid, an interesting cyclization takes place. The ketone adds to the amine to give an imine. The etamine portion adds to that imine, and it makes this spirocyclic ring system. Those compounds are interesting because they have solvatochromism properties, which is to say, as you put them in different solvents and shine UV light on it, they fluoresce at different wavelengths. And all of that was unknown to us. It was just right brain activity that got us wondering about some chemistry that wasn't in the literature. You can play fun games. You can change that from cyclohexanone to, for example, 1,4-cyclohexane dione and make these interesting dimer compounds that have the quinolinone on top and bottom separated by the cyclohexane ring. So anyway, you start playing these games. While all of that is happening, of course, the discipline side of the brain is over here making one at a time analogs that we can ship to Verkman for assays. Now, of course, we also send them these compounds, and they invariably show no activity. The fun ones generally show nothing. It's the ones that you have to just march through that actually lead to some progress. Okay, so that game sort of between Alan's assay and my synthetic lab, that back and forth takes place on this structure. While we're working on that, of course, he's continuing to screen compounds, and he finds new hits. He transfers that information to me, and now there's a new target, and we marched through that target, and that happens and happens and happens. And eventually we got to the point where we had what was a really good potentiator. So, okay, now we have a potentiator, and I finally got to say to Alan, okay, so remind me again, why is the potentiator of interest if we don't have a corrector? And he said, Mark, let me point this out to you. It's because we need this potentiator to open this channel before we can find a compound that will get the protein to the cell surface. So in other words, we needed a potentiator to modify the assay so we can now have test compounds that were looking for things that would chaperone the misguided protein to the cell surface. Does that make sense, everybody? So once I saw it, it was simple enough, but again, I spent several months wondering, what the heck are we doing? Only to find that this made sense. So it's really not a cart ahead of the horse issue. So with that assay in place, libraries are screened, structures are sent to us, analogs are made, we march forward, we make hundreds and hundreds and hundreds of compounds. And we do that to the standards of organic chemistry, which is to say we characterize them and we have all that information, looking for correctors. And so you can see here, this isn't all of them, but sort of a path of lead compounds. Eventually we found a bithiazole lead where this Y is nitrogen and X is sulfur. That seemed to be an interesting corrector. We've, for example, made all kinds of analogs of that. Here's a thiazole next to two nitrogens in the heterocycle. Change where those are, change the substituents, change the heteroatoms from oxygens to sulfur to nitrogen, all those kind of things. So there's a lot of chemistry to be done as you're poking at what makes this a good corrector and how can I improve its corrector activity. After you do a whole lot of that kind of work, make lots and lots of compounds, we found this bithiazole to be our record corrector. It's the best corrector we've found to date. So now, I'll tell you how we make that. So again, this is just one compound. The synthesis here is to start with cycloheptane dione, brominate between the two carbonyls, displace that bromoketone with a thiourea to make a thiazole, gives a ketone. We can brominate next to that ketone to give a bromoketone, treat that with another thiourea to make the second thiazole, and then and that one comes along with an R group, whatever R group we want to attach, and then we acylate that nitrogen, and that gives the final target. So that's how we made that one compound, and of course you do that same kind of thing, but with different reagents to make the analogs that you've been poking at all the time. So you end up with this compound, and here's the assay. You can see that in this assay, this compound is the top one. It has sort of 500 or 600 nanomolar activity. So it's pretty good. It's not perfect, but it's pretty good. These are a couple other ones that were pretty good, and this chloromethoxyaniline ring seems to be a constant theme. They're pretty good, but not as good. You can do fun things like, here the order of heteroatom is nitrogen, sulfur, nitrogen, sulfur. If you leave everything the same, but change the order to sulfur, nitrogen, that compound is completely inactive. So those are like, you scratch your head about that, you wonder what's going on. The interesting thing about that is it suggests something very fundamental about how these heteroatoms report themselves to the structure and to the protein that it's going to interact with. So we engaged some of our computational friends, Dean Tantillo at UC Davis, Mark Anderson at San Francisco State University. And you poke at that a bit, and we've concluded that these bithiazoles, there's a generic structure, are active when they can sort of take this U shape. And that U shape is dictated by an interesting interaction between sulfur and the lone pair on this oxygen and sulfur on the lone pair on nitrogen, which sort of sets the angle here. And then there's this odd carbon, hydrogen, nitrogen, internal hydrogen bond that sets the conformation there. And we believe that active compounds need to present in this sort of, with these kind of groups, but in that shape to be an active compound. So that's at least our thinking in the moment. So I guess I'd like to sort of wrap up the cystic fibrosis part of this by saying that this team, the Verkman lab and the Kurth labs, by putting our heads together, we're able to stare at a problem that's a pretty intriguing problem. This protein is misformed, the endoplasmic reticulum takes it apart to parts, and it gets caught in this loop. And by thinking about that project and the kinds of issues that come into play, we were able to find in a petri dish a really good corrector that gets the protein to the cell surface and a really good potentiator that caused that channel to open. Now, about 8 or 9 months ago, Vertex Pharmaceutical brought these two molecules to the market. This is their version of a corrector and their version of a potentiator. And it's a very exciting moment for people with delta 508 CFTR to find out if taking this cocktail of drugs will change this 37 to some number that we would all be happier with. So that's to be found. We were hoping that would be our molecules. It's not clear that these are the perfect solutions, so we still see some hope. But the thing that we're now focused on are what we call copos. single molecule that's both a corrector and a potentiator. So if we can find copos instead of a cocktail of drugs, maybe we can find one single molecule that will do both things, escort the protein and cause the channel to open. So that's the kind of new direction we're headed on with our CF work. So now let me change gears to the problem that people with CF live with, and that is this thick mucus on the lungs, for example, where bacteria grow. Pretty much every person, if you go to a national CF meeting, they put big placards outside of the meeting hall saying, if you're a person with cystic fibrosis and you're attending this meeting, and a lot of them do, don't come if you don't already have Pseudomonas aeruginosus infection, because people in this hall will have that infection and you'll probably get the infection. So in other words, pretty much every person with cystic fibrosis has Pseudomonas aeruginosus infection in their lungs growing in this thick mucus. And by thick mucus, that's what I'm talking about. It's a really incredible mucus. Tara tells me that she spends an hour every morning clearing her lungs and an hour every night before she goes to bed clearing her lungs. And I think that's why she's been so successful living with cystic fibrosis. She puts that very hard effort in every day. And what we wanted to do is ask, can we have some impact on that? So let me tell you the story there. This infection, this is the mucus, there's this infection growing. Our immune response by chemotaxis sends neutrophils, which are part of the innate immune system, to fight that infection. And those neutrophils are injected into the mucus. And I was surprised to learn this. Neutrophils release an enzyme called myeloperoxidase, and it produces bleach. So I said, really? We have an immune response that produces bleach. And it makes perfect sense that we would, in the innate immune response, you would think bleach is a good idea. So that's what the immune system does, is it produces bleach in this mucus to try to kill the bacteria. Now that's all good and well, except for if you have cystic fibrosis, this is a never-ending infection. And so every day you're having bleach produced in your lungs, and it leads to damage of the lungs. That damage is what led to Tara needing a lung transplant at the age of not quite 38 because of the damage done by the bleach. So we had the idea, and others have had this idea, that maybe we could help by preventing bleach formation. Doesn't address the infection. We'll have to do that with something else, but maybe we can cause some of this damage that's happening to be avoided by simply avoiding the production of bleach. So we wanted to find inhibitors of the myeloperoxidase enzyme. Starts with a library of compounds. They're screened. Now this is done with a collaborator at UC Davis Med Center, Carol Cross. He had an active assay for myeloperoxidase. So we didn't have a library of compounds. This is some work that Makhlouf and I can take credit for. We were developing the Davis-Beirut reaction. And we were making libraries of indazoles. And the literature suggested no one had tested indazoles against myeloperoxidase, but when you look at some of the compounds that were active against myeloperoxidase, we could squint and sort of see an indazole in that. So what we did was, so this is Makhlouf. Picture I took of him looking at Lake Tahoe. And if I know Makhlouf, he's not standing there with that very distinct pose of his, thinking about Lake Tahoe. He's thinking about the next substrate we should try in the Davis-Beirut reaction. This is sort of a synopsis of the Davis-Beirut reaction. The way it started with an orthonitrobenzyl bromide. Treat that with an amine, displaces the bromine to make an orthonitrobenzyl amine, and then simply treat that with aqueous KOH. And it does this very interesting heterocyclization reaction where it delivers this indazole in a two-step, very simple process. And there you go. You can make indazoles by that chemistry. Here are sort of the two generic versions of the reaction. You can start with a nitrobenzaldehyde and do a reductive amination to give the orthonitrobenzylamine. Or again, you could start with the bromide and do a displacement and make the orthonitrobenzylamine. Treat that with KOH and you can make the targeted heterocycle. It works very well on a preparative scale, 89% on a 5-gram scale. We can make these kinds of heterocycles. The thinking that we when you go back to that picture of Makhlouf at the board with the red chalk in hand, the mechanism that we were sort of contemplating suggests that there's this very interesting internal reduction of the nitro to a nitroso, internal oxidation of the amine to an imine, and it's that this reactive intermediate is what cyclizes to make the nitrogen-nitrogen bond in that interesting heterocyclization reaction to give the indazole. Okay, so you do those Davis-Beirut reactions. Of course, you can vary all kinds of things, and you start making a small collection of compounds. It was that exact collection of compounds that we sent over to Carol Cross's lab for his myeloperoxidase assay, and he reported back that we had 4 inhibitors that were nanomolar. You can see the 4 structures. That, of course, caused you to go back to the lab and make more analogs to see if you can improve on that. And we ended up finding that we could make sulfur analogs, and that the best compound to date is this 81 nanomolar inhibitor of myeloperoxidase. So I think maybe we could use that to inhibit leach production in the lungs of people with this aeruginosis infection. Okay, so now I'm going to jump to the next topic, and that's polycystic kidney disease and secretory diarrhea. And those two have a very similar problem, and that is a chloride channel stuck in the wide open conformation, chloride excreted. And you can see how that can dehydrate and lead to death by dehydration. So that's what happens in secretory diarrhea. In polycystic kidney disease, it's an interesting problem. Again, it's a genetic disease. It causes the kidney to go from a normal-sized kidney to this huge 20 to 30-pound thing that's filled with these great big water blisters. Those water blisters are formed on the surface of the kidney and internal in the kidney. And it's because the chloride channel is stuck open, pumping chloride into these little small blisters that then recruit water to solvate the chloride, and the blisters grow, and you end up with incredibly a 30-pound kidney that doesn't function. So it's a lethal disease, a genetic disease. In secretory diarrhea, it's a different story. It's an infection by cholera or heat-stable toxin, E. coli, for example, that enters the cell, messes with the signaling, and the protein gets stuck in the cell in the wide open conformation. And the World Health Organization estimates that there are 200,000 deaths from cholera toxin and 300,000 deaths from heat-stable toxin each year. 500,000 deaths, primarily children who die from this sudden exposure to one of these bacteria, and they're dead from dehydration because of this open chloride channel. So can we address that open chloride channel and can we gate it back closed? So again, it's a team. Libraries are screened. We get a hit. We make library analogs, get another hit, another hit. We ended up with this as a hit that we ended up using as our best lead, and it led to, after this kind of back and forth between the Verkman lab and my lab, it led to this compound as a low nanomolar inhibitor of the CFTR channel. So in the Petri dish, if you add that to cells, you can close the channel. So that was, I would sort of say, this is sort of the normal process as a MedChem, somebody doing synthesis. You look at the problem and you look at the structure and you start playing. And you can see from the one on the previous slide to here, we changed that to a phenyl, changed that to a furan, and we had a low nanomolar hit. Alan does other studies with these molecules. For example, in this case, he injects it into a rat and looks at the stability of the molecule. And what he found was that this compound is completely metabolized within 30 minutes and is gone. And he was able to find 2 metabolites that appeared, one at plus 16 Daltons and one at plus 14 Dalton. He reported that to us, and we stared at the molecule and thought, well, perhaps it's oxidation of that methyl group to this alcohol. That would be plus 16. And then aromatization of that heterocyclic ring, that would be a loss of 2. So plus 16 minus 2 is 14. So we thought, well, maybe that's what's happening that causes this thing to be metabolized so rapidly. So then all of a sudden the MedChem challenge changes, not to finding the best molecule, but to controlling this metabolism. What can we do synthetically to allow us to address that metabolism problem? With a bunch of new analogs being made, we thought, let's get rid of the methyl, and we found that a bromine substitution there. How do we address this oxidation? Well, how about if we make that an oxygen? Now it can't make an aromatic ring. That's neutral. So we made this analog, and if you put this as a carboxylic acid, we now have an 8 nanomolar inhibitor of the CFTR channel based on that structure. There's how that compound is made. You start with this pyridine-dione, do an acylation reaction at that position to introduce that phenyl ring and the carbonyl. Brominate that methyl to give a bromo CH2. Treat that with an amine, it gives a pyrrole. Cyclization of the nitrogen onto the carbon with the bromine and the carbonyl gives the pyrrole. And then we can do a substitution reaction at that position, of course, on the pyrrole with the targeted aldehyde to make this compound, and then simply saponification of the acid, the ester to an acid, and that's how you make that compound. Now, what's interesting about that compound is that it's racemic in this synthesis, but it only has one stereocenter. So the thought came to mind, well, what if you resolve the 2 enantiomers? Would both enantiomers be active, or what would you find? Turns out when you do that, when you separate the R and the S enantiomers, here's a crystal structure of the S enantiomer, and you do the assay, you find that the S enantiomer is completely inactive, and all of the activity lies with the R enantiomer, which is not too unusual in drugs. Most drugs that have a chiral center, you take one enantiomer because the other enantiomer oftentimes has a deleterious effect or no effect or something like that. My computational colleague just got all excited about this. And first I was like, okay, well, calm down. And he pointed out that opened an opportunity to do something that I never would have thought of doing, and that was there are not good crystal structures, but low resolution crystal structures of the CFTR protein. And he said, you know what, the fact that one enantiomer works and the other doesn't mean if I dock these 2 enantiomers into that protein structure, I can maybe suggest where it's binding to the protein, which would be very useful information. So what he did is did that. He docked both enantiomers, and he finds that sites, so there are 5 sites that seem to accommodate the inhibitor. Of those sites, the A, B, and E sites favor the S enantiomer. Well, the S enantiomer is inactive. It must mean that our compound is not binding at those sites, because if it were, we would use the S enantiomer, not the R enantiomer. Okay, so that was interesting. Site D, this site, didn't have much of a preference for S or R. They seem to both bind pretty well. And it was only site C that was selective for the R enantiomer. So our hypothesis at the moment is that our compound interacts with CFTR at that position, and maybe that's where the activity takes place. And it turns out that this is a known binding site of ATP, and ATP is part of the signaling process, so it actually makes some sense as well. So we think our compound is binding at site C. When you look at site C, and this is the R enantiomer docked and the S enantiomer, and you can just by eyeballing say, well, those are wildly different interactions between the small molecule and the protein. This is a much better fit than this. The R fits, the S doesn't. And what was interesting, too, is that you can sort of look at, well, what amino acids are around, and here's that furan moiety with the bromine at the end, and we thought, well, gee, there's a threonine and a serine. Maybe we could take advantage of those amino acids to provide an interaction with our target molecule. So what we did was we set out to make boronic acid analogs, thinking that the OH here can maybe replace the OH of the boronic acid and make a covalent attachment. This is a pretty good inhibitor. It's 40 nanomolar. It's not as good as the bromine, so we're not there yet, but I think we're on to something if we can use boron as a way to make a covalent attachment. So that's the kind of thing we're poking at in the moment. In addition to that, we're wondering, can we actually prove where the small molecules attaching or binding to the protein by doing affinity labels? So we're trying to make these, we're targeting this compound right now with a bromine, this chloronitro compound. We can, we think, displace that chloride with an azide to make an orthonitro azide, photolyze that when it's bound to the protein to make a nitrine, which would then, in the presence of the protein, have an opportunity that it doesn't have, because this is actually a known transformation from here to here. These ferrocyan oxides are made by photolyzing these nitroazides to nitrines, which then cyclize to give this heterocycle. Our thinking is that in the presence of the protein, that nitrine will find itself inserting into a bond in the protein and will make a covalent attachment. Now we can take the protein apart and look for that attachment in the data. So that's the secretory polycystic disease story. The final story is secretory is constipation and dry eye. I'm going to have to do this really quickly, so I'm just going to basically click on a couple of things here. But the point is, it's the same process, but now we're trying to open the channel, find an activator instead of an inhibitor. And how do we go about that? Well, it's the same game. You find a hit compound. This was one of the hits that we discovered, and it has that structure. It's a quinoxalenone, and so you start making analogs. You have to develop chemistry to do that. We have 3 sort of active routes to make these kind of compounds, and I won't go through all of that, but we can vary all parts of this molecule and start making analogs. We've done that. I should point out that for this case, what would be nice if you have dry eye is to put a drop in your eye that delivers this compound specifically to your eye, open the chloride channel, and take care of dry eye. That would be the plan. Now, you don't want that compound going systemically, so you want something that has a rapid, and these do, have a rapid metabolism by the kidneys or by the liver. So if anything gets into the bloodstream, it's instantly gone. That would be a good thing. So that it does what you want in the eye and doesn't do anything anyplace else. So that compound, it does that. It's metabolized in 3 to 4 hours, and Alan's rat studies all look pretty good. We've made a bunch of analogs. Here they are. The interesting thing is the best compound to date is the starting structure minus that amino group, and we go from 210 nanomolar down to 9 nanomolar by just removing that amine. And now we're in the stage of asking how's the metabolism of that, and secondly, can we also find out where this thing is interacting with the protein, and we're trying the same kind of labeling studies are underway. So we can address all those problems by MedChem, and all that remains for me to do is to thank the people that have done it. Alan Verkman is the person at UCSF who does all of the everything on cystic fibrosis other than make compounds. We do that. Dean and Mark are the computational people that I interact with and some of the things I referred to. Cross is my colleague at UC Davis in the Med Center. And of course, Makhlouf and I have been collaborating for years, and very productive collaboration, and sort of the picture of the group. Thank you.
We have time for a couple of questions. Brigitte?
Thank you very much for this talk.
Thanks.
I was wondering, at the beginning you were saying with the horse in front of the cart, regarding computational, I also see this, you make the compounds, then the computational part comes in. Do you see a slight change where you don't have to screen this large library and you have to put on your computation, your organic brain? I could change all of these groups, but I could make those, screen them computationally, and then synthesize them? Do you see it switch over in that sense?
Yeah, in fact, that would be ideal. The problem is that with cystic fibrosis, that's a transmembrane protein. CFTR is a transmembrane protein. And what I don't do crystallography of proteins, but what I'm told is that it doesn't have an active conformation unless it's inserted into the cell membrane, which precludes getting a good crystal structure. I don't know how they got the low resolution one, but the problem is we don't know where these compounds are interacting with the protein, so we can't look at a binding site and ask computationally how to change it. That would be ideal, but we just don't have that technology. And that's why we're pretty excited by, for example, this last one. If we can say, well, this is where the activator's binding, now we can take that approach and say, okay, let's throw that lead away and really just look at the site and find things that fit the site better. So I think it's something that's in the future, but we first have to find the site and get data for that site. Yeah.
Professor Meyer?
Yeah, very nice. I was interested in the channels themselves, and I gathered that iodide also went through them. And I wondered what was known about that. Are the ions solvated when they're going through, or do they strip the solvation sphere? And is there any relevance at all of this to desalinization and reverse osmosis membranes?
Oh,
Are they
that's
looking for such things?
Well, that's a very interesting idea. You know, I don't know much about that. I've actually tried to read some reviews, but I get lost in I'm a synthetic chemist and I don't quite understand the jargon being used, so I don't really know the answer. What I can say is if you bathe the cells in sodium chloride, chloride can also enter the cell. It's just that in the natural setting, it's really a one-way ticket where chloride leaves the cell. So I think that was known, and so iodide, because Verkman knew that it quenched his protein, was the reason he put iodide there, and of course it did go in. I think once the channel's open, anions can, it's just in the natural setting, it's not this direction, it's out of the cell, is how it's happening. But it's a great question I don't know the answer to, so, yeah.
Any other questions?
If you have the time, is there time?
Yes, sure.
Now, this is, first of all, a personal note. I never knew that I hold the pen to myself.
Anybody interacted with Makhlouf? Can you back me up on this?
Now I can explain it. I think it's because I don't want you to give the correct answer.
Yeah. I'm sure that's not what you're worried about.
A question. I think in the Furan system, the point is to get rid of the CH3
Yes.
or CH2, whatever. This is the problem. When you get rid of it and you put a bromine there or a boric acid, yeah, that solves the question.
Right.
Because eventually you can write a mechanism after hydroxylation whereby you get rid, you get a good leaving group in the amino or even the phenoxy. So my question is, did you try a bromo nitrogen?
Yes.
And how did that go?
Yeah, so it turns out that if you leave the
If we
nitrogen.
go to the slide, please.
Yeah, I'm not sure how quickly I can get there. Sorry.
Let's do the slide sorter.
Does that one, does that one work? At least it shows the structure. So yeah, in this structure that's going to show up here in a second,
Yes.
the bromine is
Yeah.
replacing the methyl.
You had the
and
nitrogen
an
with the methyl group.
Yeah,
Nitrogen with the methyl group.
yeah.
And my question is, did you replace that phenoxy oxygen with the nitrogen?
And have the bromine.
Yeah,
Yeah,
and
yes.
the bromine,
Yeah. Yeah, we did, and it was an inhibitor, but not nearly as good. And we think that's because once you do the oxidation and that stereocenter is lost, it really changes how it interacts with the protein. That stereocenter puts the furan in the right position. When you oxidize the heterocyclic ring, the conformation changes and it no longer interacts as well. It was an inhibitor, but only marginally when you left the...
Well, I think we know what you are going to do next summer, Dr. Haddadin. So let's thank our speaker for a very nice talk. Before we go to the coffee break, for our speakers, our guest, Malika Haj Hassan has been trying to track you since yesterday. She did interviews with 3 of you. She's going to try to do with the rest. And now we have a coffee break. We will reconvene at 11:30. The coffee break is just outside. Thank you.